So let's say you have 4 bands. You believe me that you can get densitometry with these relative amounts, right? With MSJ, it's just going to be how bright your band is. Photoshop, you have to do Photoshop or anything like that. If you believe this is bright, if I give it a higher number, right?
So you're just deciding numbers kind of arbitrarily?
Yeah, because I'm trying to explain the normalized part. And then, but this could give you relative amounts, right? Expressions, right?
Yeah.
You understand that? But you're in this situation that your starting material might be a little bit different than it actually was, but you have that data, right? How much RNA? So let's just, for the sake of that, and then let's just say you might have had.
Yeah, right here.
So which one has more RNA? Right? And so that's you just finish with arithmetic. To me, what I might do is I might do some math and say, oh, these values are normalized to a mean, something, so it might be 1, and this one might be, I don't know if this is right, 1.2 higher, but you get my math, there's a little bit more, right? So by dividing by that number, just try to find a simple. Yeah. I can help you with that. The technical part is being able to do that, and/or give the message at each time.
And then the other thing to bring that though, I used 2 different form primers.
Yep.
So can I put one guy?
Oh wow. Yep. So does anybody have any questions about how their lab stuff's going, or papers, or presentations, or anybody have any questions about anything?
Presentation. How much that we sort of all already know from being all in this class together do we want to regurgitate as context for the work that we're doing? You know what I mean?
Exactly. Well, your first thing is you're constrained to about 15 minutes. I don't want to hear all about what I've taught you and not anything about what you've done.
Right.
So, I guess minimal. I mean, I think you go in, not just, just be blunt. I mean, go in just assuming that you're talking to the class and that we know what we should know, right? You know what I mean? Because I think what you did and kind of the procedures was going to be the most interesting, and the interpretation of that. I'd rather hear more on the back side of interpretation.
That's good.
Sure.
I just quantified 2 samples of my RNA and cDNA to make sure I had something. If I don't get any other results, does it make any sense for me to quant all my samples? And does those numbers mean doesn't really?
You should focus on testing your new
Okay, so we got far out. Never mind. I didn't know that was going to happen so fast.
What's different about these primers? I totally forget.
These are for a different target, and they're species specific for Anthropo. They're for the super something dismutase.
Okay. All right, so those are for TPCR. So what band do you treat on TPCR 55? They should work. Well, you know.
Oh, my Oh, that's me. I did around this session.
All right, why are you here today?
Why?
Yes.
Just you?
Miss me?
Some
Some of you are working on your project, some of you are doing lab work, some of you are talking about your project, some of them want to talk about, want to go over review stuff too. So I'm just trying to gauge time frame. If y'all are into your papers, I'll give you a few more, I'll probably give you 2 more minutes anyway, and maybe at 2 o'clock, in 5 or 10 minutes, maybe start talking about the exam, but I want to let y'all get started on other stuff. Just tell me what you want me to do.
I'd say exam first. Go ahead. Sorry. I'd say exam.
Exam now.
I'd say we examine. Yeah. And then
Know whatever happens
go
out.
our separate ones.
Okie dokie.
What week was the first exam? First exam.
All right, what week was exam 1? Does anybody remember? What week was exam 1?
It was after xenobiotics. That was the last main topic. Week 5, maybe? Before. So right before you left.
Okay, so week, yeah, you're right. So week 4 we did xenobiotics, and then we had a little bit of review. So what's the exam is primarily going to focus on week 5, 6, 7, 8, 9, 10. Makes sense. 10 weeks. We split it in half. 5, 6, 7, 8, 9, 10. And what did we cover in week 5? We gave the general outline on the endocrine, then we talked about adrenal, and then we talked about spawning. Okay, so week 6.
talked about thyroid hormone, and then the thyroid hormones, both in vertebrates and then invertebrates. Week 7, we went on to probably a little more endo. Nope. We did that. Monday, there was a discussion that Catherine led on the noise, remember that? And then Wednesday and Friday were epigenetics. I feel like there was are we missing an endo, or did we get it after the epigenetics? Say it again? Yeah, we did. I'm just thinking about all the aspects. We did reproduction, thyroid, adrenal, glucocorticoid/adrenal. Was that it? Yeah, so Louise was one about reproduction, right? Salmon?
Like student run like noise, something like that. Is that all fair game?
Yeah. With 8, we did OA. I don't think I'm missing anything. I guess it'd be easy if we look at a calendar like this. Week 9. That was Luis's things about endocrine disruption. Adrian used the PowerPoint. Oh, and that's when I did the recording video on immunology, right? Which most of you didn't watch. That's important. Week 10? Where are we week now? Are we in week 11 now?
Yeah.
What?
First half of that week counts.
Oh, Adrian put it on. That's what you know. That was, she put it on the wrong week. That's what. So she didn't. Yeah, she did week 10, which was last week, right? Or whatever. No, last Monday was Ahmad, right? No?
Yeah.
Yeah.
And then we went
Yeah. So it was just Monday. That was Thanksgiving, and we had Wednesday off. So we didn't have one paper. Okay? No, you don't need to help me put the papers back in where they're good, because I'm not to remind me where.
Which week 10 say?
Week 10 was just the paper. That was just last week, so we didn't have class on Wednesday, and Friday was a holiday, so I'm on the paper. What was that on?
It's on what was about you guys? It was the human one.
Smoking, right.
Smoking and wine.
And cell phones, too. It was good. It provided a human context to things we taught, which you already should have learned about from the antioxidants, right? Is that what it was like? Total reactive oxygen? No, the antioxidant capacity? Is that what it was? Yeah. So those major concepts and those stuff. Okay, so let's go
All right, since we're talking about papers, who gave the paper? Who did the discussion on the hunt? Would that be Adrian? And she did the Apocalyptic Forest and ocean acidification. So that was a carryover. This week there were 2 lectures. I know. This was Catherine probably gave hers, I'm guessing, on the dolphins and sound, right? And then Emma gave her background, and then I gave the more kind of up-to-date thing on OA. Okay? I'm missing something here. There were 2 epigenetics in this week. There was something else in this week, too.
Did you say salmon reproduction or something else?
Yeah, where was the salmon? Where was Luis's talk? Luis's talk was like down here. Week 9? Alright. Anything else? There's probably another paper in here. Yeah, so Mackenzie's lecture would be this week, and then there's 2 epigenetics lectures that week. I don't remember.
Yeah, she did her overview on Wednesday, and then on Friday you guys have the paper.
That's, yeah. The royal jelly one. What was Monday of this week? That's of week 7.
The noise article? No, we did have a
Holiday?
summer.
Yes, it's kind of holiday. This is the
Or
normal.
maybe Catherine went week 7.
Okay, I just want to make sure we got the topics, and then we'll talk about the main aspects of each of them. I'm trying to remember. All right, so endocrinology in general is probably an important part to learn. Because that was, excuse me, the major kind of theme of content-wise of what we covered in 2 full weeks that carried over into the week, Louise's talk and epigenetics and ocean acidification were the other ones. What's endocrinology to study? When I say endocrinology, what do you think about? Hormones, steroids, right? Things that move from one gland or tissue to another. Other kind of general aspects that make it complex that we talked about in the beginning. What adds to the complexity of endocrinology? What's that? Yes, multiple organs, multiple target tissues. When you really think about it, you've got the receptors that come into play.
Multiple effects from the same signal.
Yep. We believe that it could be tissue dependent. We also, probably not in that first lecture, but you probably realized when we talked about adrenal and thyroid hormone, is that there was this interaction. Like a lot of the hormones were needed, or like something to be primed before they could react, before they could go up and carry out an effect. Examples of that when we talked about spawning and homing and the relationship between thyroid hormone and growth hormone and cortisol. Remember when all those things went up, that's the other part. Added to the complexity of it. All right. The first one we talked about was kind of, I put adrenal, but that's just shorthand for like the glucocorticoids and the catecholamines, kind of, because that's considered like the general stress response. And in the vertebrates, those are, you need to know like where they're produced, the axes. And all of these, there's important axes to think about. When I say axes, what am I talking about?
Signaling pathway?
Yes, signaling pathway. Oftentimes it's kind of, at least in the vertebrate system, which you should be familiar with, We talk about the signals coming from the hypothalamus and the pituitary to the target, or something to that respect. A lot of times, for example, you got thyroid stimulating hypothyroid releasing hormone and thyroid hormone, spawning. That's when we talk about luteinizing hormone, follicle stimulating hormones, CCH2, where they're being produced and they're acting on the gonads to produce steroids, whether it be testosterone or estrogen. Okay, you need to be familiar with what else. Kind of going to go all over the place here, but that's okay. What else can we talk about in reproduction? Those kind of particulars in the aquatic system. Yeah, what's that? Yeah, that's important. That's not really specific to we have that. We have to use it. Yeah, the olfactory cues, that's important. But I'm looking at melatonin, okay? The precursor. The telogen is relevant because it's important, but it's often used as a biomarker for pollution because there's environmental estrogen. Normally you get a response from the telogen, and we spent a little bit of time discussing and what you could be applying your knowledge to thinking about how you might study environmental disruption. And the telogen is a biomarker. What things in the environment like sometimes mimic estrogens? What? PDA. And then we also talk about things that are actually estrogens in the water in terms of the sewer system that are organisms. And then with your understanding of endocrinology and how generally estrogen, testosterone, and all those work, you should be able to imagine a scenario where different types of mimics could be bad for aquatic organisms. Okay? Seems like we spend a lot of time thinking about comparing and contrasting the vertebrate system and the invertebrate system. That's how we kind of call it. So what could you generally say if I showed you a picture of the giraffe and what's different about the two? Remember that lecture?
Remember the picture of the giraffe?
Yeah, so a lot of times, I think the two examples we showed in invertebrates, everything was almost happening in a single cell. Okay, I think we started out talking about kind of the hemocytes and the glucocorticoids, and I'll jump back to remember about that nice paper that kind of illustrated the fact that glucocorticoids, ACTH, POMC are effective in the oyster. And the other one was kind of, I kind of used that in terms of the telogen or in the estrogen system, how it's much simpler and compared to the vertebrate system in those two. But a lot of the molecules are similar in terms of endocrinology. So it's not rocket science, but you should be able to articulate the difference between a fish where you've got a hypothalamus, pituitary, and then a gonad, or they don't necessarily have an adrenal gland in fish where these cortisols are being produced. What does fish have that we talked about? They certainly do. But what's kind of the tissue that's analogous to an adrenal gland? Now, I knew it in class. It was the trunk kidney, the head kidney, and the trunk kidney, and it's primarily producing the head kidney. You know, you take the viscera out of a fish and look up from the bottom. That's there. What else did we talk about? Spawning. Thyroid hormone. Again, a little bit at the end of this lecture, we talked about, again, important to understand how bad things that mimic thyroid hormones could complicate things for aquatic organisms. What does thyroid hormone principally do in invertebrate organisms? Like fish, frogs, and birds. What's that? Yeah, it's in growth, primarily metabolism. Yeah, so certainly we should talk about in the flounder that was key in metamorphosis, and that's, to me, the hallmark example of thyroid hormone is its role, in the frog. That's probably what we learned in biology. And then that next lecture we talked about it in terms of invertebrates and the data that I did long ago on scallops, and again brought in kind of the relationship with the environment. Where the thyroid hormone was coming from and what were those, and then we'll go on another tangent. What's thyroid hormone? What's cool about thyroid hormone in our bodies? It's kind of the gland, your thyroid gland itself, and the fact that that space, right? Because it gets stored. And that's one of the only things that's actually stored. Iodine is necessary, you have to give some dietary supplements. There are a couple aspects of that make it unique to thyroid hormone. There was a nice illustration about the complexity of the endocrine system when we talked about the thyroid hormone, because we talked about the enzymes that convert T4 to T3 and T2, which is why I'm getting back to the scholars. That was one way to regulate it. You had to actually bind albumin that binds the thyroid hormone in the body, right? And the different receptors that you have regulated. But anyway, back to the invertebrates. When we talked about that, we talked about the different enzymes that were responsible for converting T4 to T3 of the active form, right? The deiodinases. What was special about the deiodinases? What's a deiodinase?
It's an iodine?
Yep, it's an enzyme. Exactly. So it's from a gene. So what's an enzyme made from a protein? What was cool about those proteins? The sexy element, the secus element. So they needed the selenocysteines. And so that was one reason I'm bringing that up, is because that was just another kind of relationship of how the environment. At the end of that lecture, there was a slide that showed all the different ways the environment could affect the normal process of metamorphosis. And selenium is necessary for that enzyme to work, and the selenium being uptaken by a microalgae form. Okay.
It's fair to say overall that when we're looking at these systems, looking at it through that lens of environmental stressors, environmental impacts, mimics, things like that, is like a useful way.
Useful way to go, but my feeling here is if you have a fundamental and just basic understanding of endocrinology and some of the details of the things we talked about, then you could infer everything else. Right, that's what I'm. But hopefully by me giving you a specific example how the environmental perturbs it kind of reinforces it stimulates your brain to think about how that works. But it might be a hypothetical scenario in an exam. Also along the lines of big picture, not getting too far with that in the details. I think the compare and contrast is a nice simple one. That's how I try to teach it, and we'll teach from the big picture of what we talked about in terms of both vertebrates and invertebrates, and maybe a couple of different aspects of glucocorticoids, adrenal, thyroid hormone, reproduction. less about like this part, because that was the longest guy. That was a while ago. But you actually have all the lectures, and you have the videos, and you have all of that stuff. Then epigenetics. Do you have any questions about that? We've got a whole week on that. I mean, generally that refers to anything that's, outside of the genome, or the genome that affects gene expression. It does not change the ATCP. Most of what we talked about, or what we talked about, is DNA methylation and how it acts. How does it act how does the environment affect DNA methylation, and how might that affect this? Okay, how does DNA methylation work in a fish? I'm trying to say invert in a fish. Sorry, the air is very loud. Say it again. Yeah, that's exactly right. But to get a little more specific about the specific fish, what's its function in terms of DNA methylation physiology? Why did we spend an entire week talking about epigenetics? There hasn't been relevant to physiology. What's that? Yeah, gene expression, but specifically, what's the mechanism in fish, in humans, in elephants, in gorillas, and in crabs? Okay, so in what I invert it actually regulates it as a promoter. Okay, it's all about methylation and demethylation at the promoter level to affect gene expression. Okay, demethylation, it's pretty straightforward. Classical example was, I'm not guarantee we'll talk about it. It was a long time ago, was the temperature in sex? Yeah. How temperature can affect sex ratios? I actually talked about that before. We talked about reproduction, where temperature affects DNA methylation of the aromatase gene. Okay, which aromatase is functioning estrogen. And so you saw how the mechanism and the environment how it affects reproductive physiology of fish. Okay. Just asking questions because it's not making sense.
Can you explain that? Yeah, it's weird.
Sure. I'm actually like this, because again, you're having to remember, well, you hopefully knew some of this, but you're kind of reflecting back on when we talked about the DNA, RNA, what's a promoter, and that kind of stuff. Because this is not really going to show up for people who are not here, I'm also putting it on my screen too. So essentially what we've shown is that with high temperature, this is the promoter for aromatase, which makes estrogen, okay? At high temperature, what actually happened is you got increased methylation in the promoter, It's only really only occurring in female. So I'm gonna put a bunch of M's there on the cytosines. All right. And what this does is, what would this do? This would inhibit expression of aromatase, okay, which is necessary to produce estrogen. Okay. So what's going to happen at high temperature is you're going to have an increased number of, well, let's call them genetically females, that actually the phenotype is going to be male. They're not going to be male, okay? The important part, the main, well, there's several, I'm going to say all that's important, but in the vertebrates and fish, this of how it works. And that's also a nice way to think about the environment, okay? And then it got much more complex when we talked about invertebrates, or when we came to talk about invertebrates, okay, like shellfish and other quadrupeds. Some quadrupeds don't even have DNA. For the most part, what we talked about is just that in the mammalian system, where basically all of this is methylated, and then this becomes demethylated and methylated. You've got the mosaic pattern happening in invertebrates. It's evolved to have a different putative role, which the theory being that it's involved in being able to adapt to environmental change. Okay? Does that sound familiar?
When we say mosaic pattern, that's the reason it's more
Spotty.
sporadic in stories. In vertebrates, it's more ubiquitous, or the population is pretty much across the entire time.
Yes.
It's pretty ubiquitous, but in invertebrates,
Yep.
it's spotty.
Yep. Yes, exactly. So in human fish, 90% of the genome is methylated. The promoter is kind of variable in its gene expression. And in invertebrates, you're right, it's just kind of spotty. And what is it, about 20% total? Actually, in your data, it's roughly around 20% of the genome is methylated. Okay, and it's methylated kind of all spotty all over the place. Primarily, it's going to be in genes, and what we talk about in class is that it's depending on the function of the gene, and that's what gets down to the ability to adapt. Okay?
With that mosaic methylation, it allows for multiple endpoints to transcription reading. I'm not quite understanding how that affects the adaptability of the organism to have that mosaic methylation.
Right. Okay. So I'm going to draw it. We don't know, but this is what we taught you. When mosaic over the whole genome.
Right.
I want to simplify it. What the theory is that there's genes involved in responding to environmental change. Okay? Those appear to be with limited methylation. Okay?
Okay.
Genes that are essential for function, RNA metabolism, protein metabolism, they appear, we call them housekeeping genes. We see they're primarily methylated, okay? And so what our theory is these ones that are involved in environmental change have less methylation, and what this allows for is this potential, we're looking at a genomic DNA here, correct? That in some instances, these might be introns. No, I should use all these colors. That you might get the ability to have different transcripts. Intron skipping, exon skipping, an abrupt stop site. And so one of the theories is this variability makes it that some of these transcripts will be advantageous under a particular condition, because a lot of marine invertebrates have a lot of offspring. some of them are going to die, but this kind of adds to the phenotypic background that allows for persistence and adaptation. Whereas in these, in ones that, what we see in those that are kind of housekeeping genes, they're very predictive in their nature of what's going to happen in their product, which kind of makes sense because if a gene is central to DNA metabolism, it's already more than likely to be okay? So that's how it relates to being able to adapt to environment. That's the theory of how, and how we call it, how it might help marine invertebrates adapt to environment.
What are the promoter regions like in invertebrates? They're similarly mosaic.
Yeah, there doesn't seem to be any preferential methylation or demethylation in the promoter. We actually see most, if you look over the whole genome, most of the methylation is occurring in the gene bodies and the genes and the exons and introns. And then on Friday, she talked about the royal jelly, and I think that was quite technical, but gave you a nice illustration again of how, in that particular case, how diet affects DNA methylation and how that affects phenotype.
I wasn't very convinced of the causality in that relationship.
Well, there's some questions about the causality in this too that we're dealing with, whether it's kind of beyond the class about what's in it, what's it cause and what's just an effect. It's interesting stuff because it's clearly different than what it is and how it functions in the vertebrate system. But it has a huge potential for both physiology, the organismal level, and population level, which we, again, going back to the first day of class, it's two things we want to always keep in mind.
Can you artificially methylate genes?
Yeah.
Superpowers.
What's that?
A lot of stuff.
Yeah. What else? We good with that? Ocean acidification. I mean, Emma gave you a really nice lecture about the kind of the basics of it and the chemistry. You know, the chemistry, what it is, how it affects organisms, calcification rates, organite, with the saturation level. Y'all had some good questions about that. Then I went on to talk about some of the more recent research about combining stressors and kind of, I think one of some of the take-home messages from that related to it affects metabolism, which affects their trade-offs, which is again kind of takes on as a take-home message from the class. You know, you only got a certain amount of resources available, and if you're feeling a stress that are using up ATP in the mitochondria to produce energy to maintain homeostasis, something else is normally affected. Again, we brought up there, I believe, the reactive oxygen species and antioxidants. Kind of a neat callback from the first part where what they actually were and how they produced. And in the case of ocean acidification, we're not sure, but it's likely both a direct effect. It could be a direct effect of the actual ocean acidification as a stress, producing reactive oxygen species, but it's probably also definitely a result from increased mitochondrial activity to try to produce energy to maintain homeostasis, whether it be by regulating ion pumps or trying to produce proteins necessary for calcification, etc. Anything else about ocean acidification? A lot of your papers cover that, because everybody was interested in that, right? And so what were some of the main points of the paper that touched on ocean acidification? Or maybe the noise one did, maybe I was thinking about. Well, we talked about it in terms of olfaction and Nemo, right? Right? You remember that paper? We probably talked about that in terms of reproduction and spawning, but they changed the ability of the table to find its habitat. That was a very simple paper, you know the concept of that. And then Adrian's paper looked at the coccolithic cores. And I guess Catherine's paper primarily was just noise pollution, right? Just how that affected. What question do you not want to see on the test? What's that?
Anything from week 5 or 6. Anything.
As long as it's not from week 1 through 10, right?
Ocean acidification test is all we have. Yeah.
In terms of what you know about from this class and on that ocean acidification, what are some of the themes and/or we talked about in terms of the papers, but is everybody going to die? Potentially, right? What else? Right, so a lot of it has to come down to how fast can we adapt, so that kind of ties back in to some of what we just talked about in epigenetics, which you can, consider. Also, a lot of the marine invertebrates have produced a lot of young, and although RK. Also, I think it probably came up in, at least when we talked about the Nemo paper, is, thinking about these studies in real life, these acute challenges don't necessarily represent generation over generation over generation. I mean, it is happening and it is fast, but that's the other thing you kind of think about when you're thinking about ecology and environmental change and the ability to adapt. Well, I think everything we learned in here, it really helps emphasize what we learned in class. I will not ask the question on the exam that the 3 people that are not in the lab will not be able to answer. I won't ask you what's the color of the gel box, and how many centimeters wide it is. Something like that I will not ask. But I think a lot of you're almost at an advantage with the lab because you can reinforce some of the ideas and think kind of independently about if I was to have hypotheticals kind of scenarios. Hopefully if anything, the lab, you know that proteins are different from genes because you're using different techniques to measure them, right? So that reinforces that. I would say there's other aspects of it. Yes.
Can we expect, the question on this one to be, the same depth and difficulty as the first exam?
Yep. Yeah, so it's not a comprehensive final in the sense that it's twice as long. It should take you, while you probably have 3 hours, right?
2 minutes.
It's meant to be a roughly 50-minute hour exam is how it will be written. And it's weighted the same as your first exam, right? So it focuses primarily on this material here. And it'll probably be the same as the first one. I don't know if everybody's here who didn't fully read it, but I probably will give you a choice of which questions to ask. You don't have to answer all of them. So
I would
please,
appreciate that.
huh?
I would appreciate that.
So please read the exam. The people that did answer all the questions did a pretty good job. I think they were writing pretty fast. Did you do all the questions, Jessica? I think it was Jessica and Jake did. Yeah. And Gage and I think I have to mark all. If you answered the question wrong, yeah, I don't know what I did. Nobody complains. Didn't read the instructions. You know, it'd be really fun. I won't do that thing. Like write on real fine print, like you get up and turn this in within 5 minutes without doing anything.
You should do that. That'd be awesome.
I should get up and I say, make sure you read the entire exam, all pages before you start. And if somebody can start, maybe for the last question.
Just turn it in.
Turn it in.
Or if you put like a button up top and it's like first person that touches the button. Just watch every piece very well.
I found the exam question thread, I don't know if it was very helpful last time, but we already have a quiz for this week, don't we? When we all wrote exam questions last class, and I thought that was very helpful. Going through and reading.
Yeah, so I'll do that. So we're in week 11. So technically I'm on, well maybe, it's probably good for me to maybe somebody who didn't do a quiz or quiz in time. I'll go ahead and put a quiz for week 11. Normally I do really get graded for 10, but I will ask that question so you'll have the opportunity. And as you noticed from last time, I used some of those questions because they were good questions. So that's definitely fine to do. So yeah, I'll do that today. What's that? Do you get the report evaluation, please? Any other questions about the exam?
Thanks for going over that, sir. Thanks.
And if anything comes up, just put it on the message board and I'll try to capture it and I'll put it on there. So the other thing is too is I'd like, she's going to get the course evaluations. They need to be done. I don't have to do them right now, but before you hear, you have the pen and taking it with you. But what does help me and help Claire kind of move along the class, what worked, what did not work. Fortunately, I think we're in a scenario where they have lost the lab ones. So when she gets back, I think we might ask one of the free form questions, feedback on that, because you might have noticed this lab is a little bit different than some of, well, it is probably different than some of your other labs that you've taken in terms of power structure. And so we try to improve on that and things like that. Can you open that up on the computer? You don't have to do it now, but just doing it, because some of the people aren't here, I'll give it to them at the end of class tomorrow, and the main point is to get them to get these, because these are for me, these need to be handed in to Claire, and Claire will put them, because these are confidential, I do not see these, they will be somewhere, and Claire will put them in the mailbox, but if as long as you get them, bring them to class on Friday, that's fine. If you want to give them to her now, that's fine too. So these are pretty straightforward questions that if you've taken a class before, you know. These are the Scantrons. And so, I think what we're going to have them do is demo. what if they didn't? You don't care.
I want to
So what would be the difference? You write them on the board.
maybe like what are there any aspects that you really ask for that you wish I was included? Like definitely
Could
what
be
we
two questions. What aspects of the lab?
instead of class and lab.
Write this one on the board.
You guys got this? I'm erasing it.
Okay. So these are for reports, not the lab. This will be a page being actually provided with here to not me, not visual, so pretty much we're going to You also have actually more.
It's a block.
Actually, more beneficial to me to these forms, okay, where you write something, okay, these are what I read to approve the class. It's going to get a little bit tricky here, okay, all right. These were the class. Okay. On the back of this sheet, this blank, okay, I'm going to answer two other questions because our administration involves the lab evaluation. So minimally, I'd like you to answer the two questions on the back of this. What aspect of lab contributed most to your learning, and then what suggestions do you have for lab? Okay, you can write it on it. But everything on this side will just be for the okay? That way we can ensure we get some written feedback about the problem.
Just went over some topics for
So if you guys could answer these just on the back, like Steven said, and then the front is just for Steven's class, not labs not included. They lost my